The focus of this video is going to be covering as much as possible of this particular video and the subsequent ones. This is a video intro to neuroscience by Neuroscience Online, delivered by John H. Byrne. He is a PhD for the medical neuroscience course at the McGovern Medical School at UTH Health, Houston, Texas. I have seen some portion of this video already, which deals with different terms and like introduction to neuron once again and action potentials and stuff of that nature. For instance, they like included, I think this is a machine that measures electricity and electrical signals. So I think here we are inducing an electrical signal or measuring it perhaps. I think we are measuring it to see what reading we get. And this is a neuron, ideal neuron. And we have also seen how we can like induce a signal electrically using a battery. I can go full screen. Yes, using a battery and like stimulating electrode. We are stimulating here. I have to know though few questions. Number one, why is a positive sign here, and how are we inducing it? And we are recording the electrode. I think we're getting a voltage and depolarization. I think the concept of depolarization is important as well, because once we have a threshold for no, let me take a step back. We can stimulate current, and what is going to happen is it's going to build up but not quite reach the threshold, but certain amount and certain quantity of signal intensity, I guess, is going to drive it over the threshold. And I think that concept is known as depolarization. Again, I have to do depolarization and action potential is the name. Okay, I have to do a background reading once again. I've seen but not quite grasped it, so maybe when I run it through ChatGPT, it can like summarize it for me better. Then we see different that we can,
This is your brain.
well, going to, it's just we see that when we can generate basically multiple signals. So this is an animation. Wait. Yes. So we have this, not quite, I think, passing here. There's like one signal, and this is basically mapping onto the reality of stimulus. For instance, when we get stimulus on our hand, let's say there is like light hit, and the difference between a light hit and a fire. If we have a light hit, we might not register that in our brain, but we have like overwhelming amount of stimulus, intense amount of stimulus, we are going to register that in our head, and that is basically like signal, 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 signal. That is the extent of that.
Changes. Okay, now, with that very brief introduction into the anatomy and physiology of nerve cells, we can start to talk about how nerve cells are connected to each other. And there's two basic flavors of the ways in which neurons talk to each other. There's a so-called excitatory connection, and there's a so-called inhibitory connection. So let's talk about the excitatory connection first, because it's the most obvious. So here's a cartoon of two neurons, a presynaptic neuron, this one nicely coded in green. We're going to call this an excitatory neuron for reasons that will become clear in a moment. And that neuron is making a synaptic contact with a postsynaptic neuron, this blue one. And we're making intracellular recordings, as shown below here, from both the postsynaptic neuron, that's the blue trace here, and the green excitatory neuron. Now, at this point in time, an action potential is initiated in the excitatory neuron. We don't have to worry for the moment how that was initiated, perhaps through a intracellular injection of current or perhaps by some sensory stimulus. But what you see here, importantly, is the action potential in the presynaptic neuron and the consequence of that action potential. There is a potential change in the postsynaptic neuron. And this potential is a depolarization. The membrane potential has become less negative. Such a potential.
Okay, so I think I have figured it out what it means to depolarize. So I think the inside of the cell is negative, charged with negative ion, and the outside of the cell is much more positive, so it has more positive cells. So when we induce it, and that's why like it was the key to like why was there a positive, yes, I think this portion, why was there like the positive end attached to the cell. So the idea is that the positive charges are going to enter the cell, and it is going to make the cell depolarized, and that is how the signal or electrical pulse is going to be translated from one place to the other.
Is called an excitatory postsynaptic potential. Excitatory because it depolarizes the cell, bringing the cell towards threshold. And postsynaptic, for obvious reasons, it's a potential recorded on the postsynaptic side of the synapse. Now, in most cases, a single action potential in the presynaptic neuron, while it produces a depolarization of the postsynaptic neuron, that depolarization is insufficiently large to reach threshold and trigger an action potential in the postsynaptic neuron. But if two of those action potentials are fired in quick succession, the EPSP produced by the second action potential adds to the EPSP produced by the first action potential, and now that summated EPSP is capable of reaching threshold and firing an action potential in the postsynaptic neuron. So now with this simple circuit, we have allowed one neuron to activate another neuron, and that's a go signal, if you will, in a very simplistic manner for propagating information through this circuit. Okay, let's talk about that other flavor of synaptic transmission. This is the inhibitory synaptic transmission. So the top you see the same excitatory neuron that we talked about earlier. The bottom now shows this red neuron, which, for obvious reasons, we'll soon see is called an inhibitory neuron. Now, the first part of this panel is identical to what I showed you previously. An EPSP produced by the excitatory neuron by a single action potential produces a subthreshold EPSP. Two together produce the summating EPSP, leading to an action potential. So here's the inhibitory interneuron. Now we produce an action potential in the inhibitory neuron, and what do you see in the postsynaptic cell is that the membrane potential becomes more hyperpolarized, more negative, which is called a hyperpolarization as opposed to a depolarization. So as a result of the chemical transmitter substance released here, the membrane potential of the postsynaptic cell becomes more negative. What's the functional consequence of that? Well, when you just look at this, you say, well, it doesn't do anything. The neuron wasn't firing any action potentials to begin with, and it's still not firing any action potentials because of the IPSP, inhibitory postsynaptic potential. This is called an EPSP, or excitatory postsynaptic potential. So what good are these IPSP?
Okay, EPSP and IPSP. Just took notes.
Somebody is saying they reduce the probability of initiating an action potential. Absolutely correct. And you see that. You see that when you challenge the system with a combination of excitatory and inhibitory input. So the IPSP, it hyperpolarizes the cell, decreases the probability of the postsynaptic cell from firing. Now here is, you see, this process of integration. Here was the IPSP produced by the red neuron. Now you have another action potential in the red neuron producing the IPSP, but shortly thereafter you produce the same two action potentials in the excitatory neuron that you produced over here. Over here, these two action potentials produce EPSPs that summated to reach threshold and produce an action potential. Over here, the two EPSPs summate, but because of the simultaneous inhibition, now the summation fails to reach threshold, and the postsynaptic neuron does not fire an action potential. So this inhibitory neuron can regulate.
Also confusing to me is who is doing the firing. So is it the presynaptic neuron or the postsynaptic? Okay, so in this context, I can see why the postsynaptic, like this is the subject matter. Like these two presynaptic neurons are basically working on the post one. So I understand. Makes
Can
sense.
regulate the ability of the excitatory neuron to drive the postsynaptic neuron. Or, as was said by one of the students, it can regulate the probability of the postsynaptic neuron being active. Okay, now let's talk about micro-network motifs. Fancy word, but all it means is that despite the tens of thousands of different ways neurons can connect with each other, there's a certain number of common motifs that occur very frequently in neural circuits used by the nervous system. And what I've illustrated here is some of those common motifs that you're going to be seeing over and over again throughout the course. Let me just give you a very brief introduction to them, and then we'll talk about some of them in more detail. So here is feed-forward excitation. We've already seen that. One neuron excites a postsynaptic neuron. Here is a circuit called feed-forward inhibition. In this diagram, by the way, the green cells are ones that have excitatory effects. The red cells have inhibitory effects. So this one's called feed-forward inhibition. An excitatory neuron activates this red neuron, and this red neuron, in turn, inhibits the neuron that it's connected to. It seems kind of odd, and we'll see how this actually works in a real circuit. And then there's this phenomenon of convergence and divergence. And it's
Language matters. So he is referring to these as circuits. Um, I think earlier, probably this is the lecture. He compares the brain with a computer network, as opposed to the brain itself being a computer. That might be low resolution analogy to compare brain with a computer. A higher pixeled one would be to imagine or picture the brain as a network of computers, each doing special complicated tasks. So I think the language game here matters. The comparisons or the mode of comparisons are tech and computer alike.
is a restatement of what I told you about the basic connectivity patterns. So here you see one postsynaptic neuron receiving connections from 3 different neurons. So these 3 different neurons are converging on the same postsynaptic neuron. So here's convergence, and here is divergence. One individual presynaptic neuron connects to many different postsynaptic neurons. Here's a circuit called lateral inhibition. An excitatory neuron connects or excites an inhibitory neuron, and that inhibitory neuron in turn feeds back to inhibit adjacent neurons to the first one that was active. I'll show you more details of this in a moment. Here's an interesting circuit called feedback or recurrent inhibition. An excitatory neuron activates a postsynaptic neuron. The postsynaptic neuron activates an inhibitory neuron that makes an inhibitory connection onto the neuron that was first activated. Does anybody see a possible function for this, just thinking about it? It's kind of like a
No.
braking mechanism. This neuron gets active, the postsynaptic neuron gets active, the postsynaptic neuron feeds back and says, hold on, I don't want you to be active anymore, I had enough. So this is a braking mechanism. Once you start this thing going, it comes back and turns it off. Here's an interesting circuit. Look at this one. An inhibitory neuron connects to an inhibitory neuron, which connects to another inhibitory neuron, which connects to another inhibitory interneuron, which connects back to itself. What could this possibly do? We'll see in a moment. And then there's, just like there's feedback or recurrent inhibition, there's feedback or recurrent excitation. And what do you think intuitively this kind of circuit can do? It's a switch.
It's just feedback. Okay.
Once this neuron is active, the postsynaptic neuron is active, which then feeds back to activate the neuron that started it all. So once you get this thing going, it will go on and on forever, in principle. So this is a switch. And switches are really important in some cases in the nervous system because you'd like to have some of your neurons switching on right now, the neurons that are involved in memory mechanisms, so you can remember this lecture tomorrow. So let's talk about some of these in more detail. Let's talk about the feed-forward excitation and feed-forward inhibition, and this is particularly relevant to what you just heard from Dr. Oakes. So here is the stretch reflex, the monosynaptic reflex that Dr. Oakes talked about, because it's called the monosynaptic reflex because it involves a single synapse in the spinal cord. It's actually a little more complicated, but basically it can be boiled down to that. So here's that neurologist hammer that Dr. Oakes talked about. The hammer taps the tendon, stretches the tendon, activates receptors, stretch receptors. Those stretch receptors initiate action potentials indicated by these little circles. The action potentials then propagate to the central nervous system, to the spinal cord, where they produce a feed-forward excitatory connection onto this blue neuron. That neuron becomes active. Action potentials propagate out the ventral root to cause the contraction of the skeletal muscle, and here's the subsequent reflex. This would be a grade 2 reflex based on your chart. Simple monosynaptic reflex involving a feed-forward excitation circuit.
My question would be, is it just one neuron doing the trick? Like a elongated neuron stretching from the leg all the way to the brain, which strikes me unlikely. Or what should be the natural thing that is like a sequence of neurons, and pre and post synaptic neurons stretching all the way to the brain.
Now, what you also see here is another interesting circuit motif that we've talked about. Look at this neuron here, which is called an interneuron, this black one. Now, an interneuron is called an interneuron because it's interposed between one neuron and another neuron. So here, the interneuron is interposed between this blue sensory excitatory neuron and this red motor neuron, which happens to be a flexor motor neuron, right? Extensor motor neuron, flexor motor neuron. Note that with this reflex, we activate the extensor motor neuron. Isn't that what you want to do, to have the reflex? This is, by the way, as was indicated, this is a simple way of assessing the integrity of the nervous system with this simple reflex test. All right? So it tells us how well information is being propagated here. It tells us something about synaptic transmission. It tells us about the motor synapse here in the muscle cell. So one can glean a lot of information about the integrity of our nervous system just by using this simple reflex and others. Okay, the neuron, the presynaptic neuron here, the green one, excites the interneuron. The interneuron releases the neurotransmitter producing an IPSP in the flexor motor neuron. Why do you want to inhibit the flexor motor neuron? Because you want to get the extension, you want to decrease the probability of having this neuron being active and producing an inappropriate contraction, right? So this feed-forward inhibition is a way of regulating neural circuits. Let's talk about convergence and divergence using, again, the spinal circuit. So here is the very simplified version of the basic circuit. The sensory neuron connects to the extensor motor neuron. It also connects to the inhibitory interneuron, which in this case would inhibit the flexor motor neuron. It's more complicated than that, is that the sensory neuron just doesn't connect to one motor neuron, one extensor motor neuron. It connects to many different extensor motor neurons. So this is the principle of divergence. And here's the principle of convergence. Any one motor neuron doesn't just receive synaptic connections from a single sensory neuron, it receives synaptic connections from many different sensory neurons. So it's this combination of convergence and divergence that really makes this reflex work. It's not just that one synapse, it's a whole bunch of synapses. Okay, let's talk about lateral inhibition. Lateral inhibition is a very important circuit that mediates a phenomenon.
I don't think I can summarize what I just saw, but if I had to try, I would say that our muscles could pick up a great amount of sensors or sensory inputs, stimulus, and the responses also vary greatly. So, for instance, there is a different response to just maybe a ant bite versus something very severe in intensity or pressure. Like say someone beats you up with a hammer, so the reaction is going to be different. The pain threshold is going to be different, and obviously our brain can like alter the response accordingly.
Called edge enhancement. And what I mean by that is shown in this little panel here, showing two rectangles, a dark one and a lighter one next to each other. Now, if you look at this carefully, I can tell you in advance that the illumination here, the intensity of the illumination is totally uniform throughout, and the intensity for the darker rectangle, and it's totally uniform throughout for the lighter rectangle. But if you
Uh-huh.
look closely at the border between these two rectangles, you may notice that at the border of the darker rectangle, it seems a little bit darker than the rest of the rectangle. And at the border of the light rectangle with the dark rectangle, it seems a little bit lighter here than it does throughout the rest of the rectangle. So this is does everybody see that? This is not a trick. This is totally the same illumination here and here. So this is edge enhancement. This allows us to detect edges, and that can be really important if you're walking at night near a cliff. You want to make sure you know where the edge is, right? So edge enhancement.
It also comes into play on how we detect images. So, for instance, AI or any of the softwares use some form of matrix to identify different grids. I think the idea is that you have like different matrices and you assign different grid numbers and you determine the edges of different things based on that. So, it should be like if we are looking at this something I think the word gradient came into play as well. So if you're looking at this particular area, it's going from one color to a common shade, perhaps common shade to this at the edge is going to be a radical shift to the next thing. So that is how I think AI understands color in general.
How does this work? This simple circuit of lateral inhibition allows us to make this distinction. And here's a very simple cartoon of how it works. So here's the dark rectangle, here's the light rectangle. Now, and here's a very simple description of the circuitry in the retina. So we have light impinging on photoreceptors, and as a result of the light, the photoreceptors become active and initiate action potentials. Those photoreceptors, in turn, make synaptic connections to so-called second-order neurons, and those neurons then transmit information to the central nervous system, right? And the assumption here is, so these fives, see the 5, 5, 5, and 10, 10, 10, that's just a reflection of the intensity of illumination. Let's just say this is 5 lumens here, and the brighter structure is 10 lumens, right?
Lumen.
So because you
Is
have
lumen the unit of light?
lumens, we're going to make a very simple assumption that an intensity of light of 5 lumens produces action potentials of 5 hertz, or 5 per second, right? So 5 per second here, 5 per second here, 5 per second here, and the neurons that are activated by the brighter intensity stimulus to the retina then fire more action potentials. So 10 lumens, this is a great simplification, fire 10 action potentials per second. So if this was all there was to the circuit without lateral inhibition, what you would perceive is exactly what is presented to the eye, right? This is dark and this is bright, and there's no difference at the edge. Now, with lateral inhibition, things change. So here's the lateral inhibition. Each one of these retinal receptors makes an inhibitory connection to adjacent downstream second-order neurons. The connection is weak, as indicated by this point 2. The normal excitatory connection is +1. The inhibitory connection is -2. So each one of these neurons makes an inhibitory connection to its neighbor. That's the lateral inhibition. Now, note the consequences of this. Over here, away from the edge, the 5 intensity of illumination now produces fewer action potentials because there's some inhibition from the neighbor. But it's still 3 here and 3 here, right? That's fine. And the neurons that are responding to the brighter illumination, they are still firing at a constant rate. It's somewhat reduced, 6 and 6. But the real interesting thing occurs here at the edge, because now the neuron that is receiving the illumination at the edge receives less inhibition from this neuron at the edge, and therefore its output is greater because it's getting less inhibition from its partner over here. And this neuron, which is responding to the dim illumination, is receiving more inhibition from its neighbor to the right in the bright region. So the net consequence is that the neuron that's responding here is firing less than its partners, and the neuron that's responding here is responding more, giving a perceived light difference at the edge. Think about this. It's not intuitively obvious, but if you work through the numbers, I think it will become clear. Now, lateral inhibition.
It is clear. I think this is nice because it is explaining why we have discrepancies in how we perceive things. So what comes to mind is like optical illusions and why they are formed. This is a good um, demystifying piece of explanation which basically explains how things are and why we, so for instance, this is like close to an optical illusion where we might see, not this one is not doing that trick, but there are like versions of it which a static image you can see and perceive that to be moving. So I think this one, this portion of the video demystifies that for me and makes it explainable, which is also nice.
Great, because it provides us with this edge enhancement. But it also has a problem, and it leads to a so-called visual illusion. And there's one very famous visual illusion called mock bands. You've probably all seen this again. So here what we see is a gradient of illumination. See, it's bright, dark, bright, dark, bright, dark, bright, dark, right throughout. It's all obvious. And also what you see here is a horizontal stripe. And you'll note with this horizontal stripe that it appears to change in illumination. It's dark here, it's bright here, dark here, bright here. Does everybody see that?
Uh-huh.
Well, you're all seeing an illusion because it's the exact intensity here as it is here. And you can see that by putting a mask over this. So you don't see this anymore, you just see the horizontal bar. Here we go, here's the mask. And there's no tricks here. There's the mask.
But also it
Exact
looks a
same
shade darker.
intensity. So you've all been tricked. And this shows that what you think you see is what sometimes is not really there. This is just a simple illusion. How does this work? So the property of lateral inhibition explains this. Why is this dark here? This region is perceived as being dark here because the neurons that are responding to this intensity in the retina have neighbors that are responding to a bright light. So they are inhibited by their neighbors. And why is this appear to be darker here? Because this region is dark, so these neurons are inhibiting the neurons responding to the band less so, right? So there is your visual illusion. And just as the lateral inhibition can explain edge enhancement, it can also explain this very ubiquitous visual illusion. Let's talk about feedback or recurrent inhibition. And before I talk about
Which
in
one?
terms of neural
Which
net,
one? Which one we studied? Lateral inhibition. Which one did convergence, divergence, lateral inhibition? I think this one.
or in my
Example, this one we just saw. We saw this one. We saw this one. This like feed forward excitation, feed forward inhibition, and convergence, divergence, basically explaining, in my words, not his, and I might be wrong, on how our body could respond to varying degrees of stimulus, and the response could be varying degrees as well from our side of the brain, the output basically. And then we learned lateral inhibition, not inhibition, but inhibition. Now we're going to learn feedback recurrent inhibition, feedback slash recurrent inhibition.
network, I want to talk about it in terms of a so-called nano network. So there's three levels of networks that we're going to be talking about. A micro network, that's these types involving a small number of cells. A nano network involving biochemical reactions in networks. And finally, macro networks that involve large connections or collections of micro networks. So a nano network of feedback inhibition is important
Did the
for
audio go
producing
up?
a phenomenon as shown here. Now, the standard textbook version of neurons is either they are silent or they fire action potentials when they are stimulated with a synaptic input. But some neurons, like this one, have a so-called endogenous bursting behavior. So in the absence of any input, this neuron is firing bursts of action potentials, becoming silent, firing bursts of action potentials again, and becoming silent. So this is called an endogenous bursting activity. These kinds of activity patterns could be important for rhythmic behavior, like respiration. Well, how is it that we breathe in and out? This kind of neuron could be responsible for those kinds of rhythmic behaviors. How does this work? This works through a nano circuit. Here is an idealized version of that neuron, the outside of the cell, the inside of the cell. These cells have specialized channels that allow for calcium influx to occur.
Another, yeah, not just another, but two set of words. One is an influx, another one is a
These cells have specialized
channels. Yes, the word I was looking for. Channels is something that comes up in, I think it started with the first lecture I saw Professor Fink, and also subsequent lectures of Robert Sapolsky. The word channel comes up a lot when talking about neurons and how chemicals outside, mostly ions outside of the cell, enters in. So the term for that is channel. And the word influx is a good vocab to keep in your repertoire.
channels that allow for calcium influx to occur. Calcium is a positively charged ion. It's at high concentration outside, low concentration inside. The feedback inhibition network, and I want to compare this network with this network. So the calcium comes in, and when calcium comes in, it depolarizes the cell. And as it depolarizes the cell, it brings the cell to threshold, causing the initiation of action potentials. But the interesting part here is that the increase in calcium also reduces the conductance of this channel to allow calcium inflow to occur, so the calcium inflow stops. But that calcium then, which is inhibiting the inflow of calcium, then is taken up by buffers, and so the calcium concentration is reduced, allowing the influx of calcium to occur again and repeating the cycle. So the point that I'm trying to make here is that this type of feedback inhibition circuit is capable of generating an oscillatory behavior in a neuron and an oscillatory behavior in a nerve circuit.
Oscillatory, okay. Basically, repeating and doing this thing, doing its thing, and then like receding away without any issue.
It's a relative.
Not any issue, that is poor vocab. Without any active mechanism, perhaps like active push from the brain, like there's like no stimulus sent. It is like by default it enters, and once it enters, it basically lowers the entryway, or like it makes it smaller, so that less is a less amount of that substance can come in. Substance maybe is an ion, so less amount of that ion can come in, and then the ion itself vanishes away, creating a need to have more of that ion.
Short-lasting type of phenomena, short-term dynamics, once every 10 to 15 seconds. There is a subcellular network that's due to feedback inhibition that underlies 24-hour rhythms. And the big 24-hour rhythm that you all know about is the circadian rhythm. Mm-hmm. Circadian rhythms are due to oscillations produced in a special part of the brain called the suprachiasmatic nucleus. It's located just above the optic nerve. And what the suprachiasmatic nucleus does is it generates rhythmic patterns of output. And those rhythmic patterns of output have widespread effects on the release of hormones from the pineal gland, melatonin, regulation of the autonomic nervous system, the daily cycle of our body temperature, regulation of hormonal release. Now, it's interesting that this very complex and profound phenomenon can really be boiled down to a simple feedback inhibition circuit among one gene. So there's a gene called PER, and PER leads to the synthesis of the PER message and, in turn, the PER protein. PER protein then feeds back to inhibit the transcription of the gene. And this inhibitory cycle has a time course of about 24 hours. So the protein is made, it then inhibits the cycle, the protein is degraded, relieving the inhibition, allowing the cycle to happen again, making more protein, inhibiting the transcription, the protein is degraded, then the cycle repeats itself over and over again at a 24-hour period.
Degraded, the term is degraded. I said like vanishes away, but the word is degraded.
What is sustainability?
Remember that crazy circuit with the 4 inhibitory neurons inhibiting each other? Well, that can explain, at least in principle, quadrupedal locomotion. So here's a little doggy showing the pattern of a walk. It's a gait, so-called gait, right? And a walk is associated with a movement of the left front limb, and quarter of a cycle later, the right hind limb, and then half cycle later, the right front, and then the left hind. So the question is, how can this kind of gait be produced by the nervous system? Can a simple circuit do it? Another secondary question is that animals can do more than just walk. As shown over here, they can also trot. A trot is a gait associated with in-phase movements of the left front and the right hind, and with 180 degrees out of phase of the opposite limbs. But they can also do something called a bound, where the 2 front limbs are in phase and 180 degrees out of phase with the rears. And then there's something like
called
in phase limbs. But they can also do something called a bound, where the 2 front limbs
The
are in
bounding.
phase
Okay.
and 180 degrees out of phase with the
Leaps and bound.
movers.
Oh, okay. So leaps would be jumping and bounding would be like, okay,
And there's something like a gallop, which is very similar to a bound.
gallop.
So we don't understand the neural circuits that produce these, but let's think about ways in which, in principle, you could do it. There's all the dogs. You can get these animations online. So one way you could do it is as follows. Remember that endogenous bursting neuron? You could take 4 of those endogenous bursting neurons and have one neuron hooked up to the left front limb, another hooked up to the left hind limb, and so forth. And if you set up the timing just right, so you put these 2 guys in phase with each other, and these guys, hind limbs, 180 degrees out of phase with the first 2, you could, in principle, produce a gallop. These 2 limbs would go first, and then would be followed half a cycle later by the hind limbs. Right? In theory, it would work, but in practice, there'd be problems because one is you'd have to set these up so they're exactly in phase, right, like this. And what would happen if there was some drift in the activity? So this one started firing a little bit earlier or later. Well, your problem would be that things would get very uncoordinated. So this doggy is not going to gallop. In fact, it's not even going to walk very well. So one way you could do this is with this circuit here. You take now 4 of these neurons, and each one becomes a bursting neuron. And when you do that, you get a circuit like this. And if you do a simple twist of the circuit, shown here, it's the same circuit. This blue one, that was that green one down here is now up here. Now this circuit, with just those 4 neurons, with those endogenous bursting properties, can produce each of those 4 different gallops with slight modifications in the strength of the synaptic connections between the neurons. So here's an example where this relatively simple circuit can, at least in principle, produce a fairly complex behavior, which gives us hope for understanding more complex behaviors in the nervous system. Here's a circuit I'm not going to go into. You can read about it. This is a circuit for the recurrent feedback excitation. This is important in the hippocampus. That's a region of the brain important in memory mechanisms where the memory is distributed among the strength of the synaptic connections. And then finally, we have macro circuits. All these different regions of the brain are highly interconnected to each other. And within each one of these regions are microcircuits, but this microcircuit is communicating with another microcircuit over here. And collectively, these circuits form something that looks like this. This is a macro circuit for the processing of visual information. You need to commit this to memory. No, I'm only joking. But just to illustrate the point of how complicated things are. So here, down here, processing of information from the retinal ganglion cells to the lateral geniculate up to various visual cortical areas. All these different cortical areas are communicating with each other through feed-forward and feedback connections. We've given you a brief introduction to the properties of neurons and neural circuits. What you're going to learn about next in the lecture from Dr. Bean is how these circuits are formed. How do they form in development, and how is it possible that one neuron can be specifically wired and connected to its appropriate partner. Okay, thank you.
Okay. My feedback on the lecture. So, I liked for the most part because of how simple it was to follow, and it was addressing very basic stuff that I hadn't previously considered. So that is like a new form of learning, especially how much similar or alike the brain is to a circuit. So I've seen circuit in my secondary higher education, and there it can be I know, right? I think the circuit is a good comparison, but also the comparison of logic gates, basically. Logic gate, I think, is a good comparison, and the circuits obviously use logic gates. But in the ICT studies, I think I remember from that the logic gates, and most of these lectures can be viewed as like how the brain reacts to or acts very similar to that logic gate. So that is cool and that is neat, and we also learn a few of the processes that we carry out on a regular basis. How I think the best thing about this is how scientists or scientifically those things are broken down into small micro structures. To think about, it's not as simple as okay, the brain does this, brain does coordination, and that's like okay, nothing else to study. No, it is further broken down into very complex chains to understand how it is working. So for me, I think I understood so far thus far. The exposure for me was with respect to like two very micro cells, neurons, interacting with each other, and now I'm seeing how the interaction between two cells can create a microstructure, and microstructure can like create microcircuit. I'm going to use the word circuit, and a bunch of circuits can come together to form a large complex circuits as well, which help us carry out like significantly complex things. uh, and not just like we can carry out those things naturally, but these things help us actually visualize, no, actually map out how mechanically we can do those things. So for instance, the use case would be if we're building AI, this is the model we're going to use. This is how I think they're going to be modeling the AIs and robots to mimic how human mind and the chemicals inside the human mind operate. So with that being said, I think it's fair. This is like a bit too much on the technical side as opposed to, and perhaps this is because that's how the neuroscience subject is taught. And so far I have not done a concentrated neuroscience course. I am like physiology and like introduction to human behavior from Sapolsky, or evolutionary human behavior. I am forgetting the name of the playlist. That was in neuroscience in its full glory, and this is neuroscience in its full glory. So with that being said, I think I'm going to stop recording now. And if I do a second, if I watch the second video, we can record that later.